Now, if I want to define lipids, how will you define lipids? For example, so far, if I have to define carbohydrates, we'll say that these are made up of sugar units and they have a glycosidic bonds. So you have a polymer which has a sugar unit as a monomer, and you have glycosidic bonds. If I want to define proteins, what we have is that we have amino acids as a monomer, and the polymeric form of amino acid having peptide bond can be called as proteins or polypeptides. Same way for nucleic acids or polynucleotides, you can say that these are polymer of nucleotides having phosphodiester bonds. Can we define in the same manner these lipids? And if yes, why? If not, why? Can you think of, imagine a couple of structures of lipids and then try to define them? What is your general concept of lipids? What are lipids? Can you name any lipid? You are, aware of oil. You are aware of ghee. You are aware of fats, which is accumulated inside the body. So what are they? These molecules or these structures are basically triacyl glycerols. We will talk about the structure more. But what you find is that the lipids, they cannot be defined just based on the bond. Though they will have bond, they are mostly, you will find that there will be ester kind of a linkage between fatty acid and OH group of either glycerol or other structures. But if you want to define lipids based on their structure, it is very difficult because they are variable in structure. Their structure varies from one form to another form. You have on one hand, you have a triacylglycerol, on the other hand, you have a cholesterol, which has a lot of difference in their structure. And that's why we do not define lipids based on their structure. Rather, lipids can be defined based on their solubility to the hydrophilic solvents and hydrophobic solvents. So what happens, the lipids are molecule which is present in the biological system, whether it is a animal cell, plant cell, or microbial cell, and which is soluble in hydrophobic solvent or organic solvent, and insoluble in hydrophilic solvent like water. So these are the structure which can be defined based on their solubility. Is it clear? Slowly you will see the different structures and then you will appreciate that their forms are very variable and they can range from a very simple structure to very complex structure. Now looking at their specific role in a membrane, you will find that they are part of a biological membrane. So the boundaries of the cell membrane, they are formed by biological membranes. So suppose I have a cell and I want to define what is the boundary of the cell. So how you define the boundary of the cell is defined by a membrane. And what do we call this membrane? This is called as a plasma membrane. Other than that, you will also have a boundary of different organelle. For example, nucleus. Nucleus is bound by nuclear membrane. You can also have a Golgi body, which is also bound by, membrane. So you have internally several types of membrane which is present. You can have a mitochondria. Suppose you have a mitochondria, you will again have a membrane, which is, double layer membrane. Chloroplast, that is having membrane. So there are many intracellular membrane as well. Because most of the membrane, they have some kind of a property which matches with each other, and that is why we club them together. And here we are going to talk about the membrane, which could be plasma membrane or mitochondrial membrane or nuclear membrane. We are not differentiating based on their location, but we will give you the basic property of this membrane. So what is the property of this biological membrane? First fundamental property of the biological membrane is that they are selectively permeable. So they have shown their permeability for something but not for others. So they will be selectively permeable. So the question comes how they become selectively permeable. So we'll talk about that. What makes them to be selective permeable? Then the lipid component of the membrane form the permeability barrier. So the lipids, they are hydrophobic in nature by their definition, and that is why they form permeability barrier. So what kind of a permeability barrier? Are they the barrier for every molecule, or they barrier for a specific type of molecule? So you will learn that the membranes, they are barrier for hydrophilic molecules, except for one of the exception, which is the water. I'll show you how they act as a permeability barrier. Then, as I said, the membrane, they are not only lipids, they are also made up of proteins. So these protein component, they are the one which act as a transporter. They are the one which carry the molecular side. They can act as a transport system, they can act as a pump, they can act as a channel. They are the one which are responsible for carrying the molecule from one part of the membrane or one side of the membrane to another side of the membrane. Another important feature you will learn, yes, such as storage of energy as well as information transduction, that are dictated by the proteins associated with them. So if I look at the membrane and if I talk about what component is playing a major role, basically the lipids, they are the permeability barrier, whereas the proteins, which are a part of the biological membrane, they are the one responsible for energy transfer, they are responsible for different functions. So the proteins are the component which are playing most of the role, even the transport process, whereas a lipid acts as a barrier. Now here I have given you a structure of a soap bubble, which gives you some kind of idea that how lipids looks like. So I take one of the lipid molecules, see this one. So what you have is that you have a circular shape and there is a wavy structure. So what does it mean? So this part makes hydrophilic, and the rest part of this structure, this part is a hydrophobic. So the green one is hydrophobic and the red one is a hydrophilic. We will talk more about how it works out, what are the components actually in the lipid. That part we'll discuss in next few minutes. But giving you one of the very clear example where such kind of a structure by the hydrophilic as well as hydrophobic, these are called as amphipathic, which is present in the soap bubble. And um, it forms a bilayer kind of a structure. Let us move on. How membrane looks like. I'm taking example from bacteria, which you know. So these are examples which are giving you some idea about the lipid. And to just talk about these lipids, what you have is in some of the bacteria, you have two types of membrane. One is the outer membrane, another is the inner membrane, and in between you have a cell wall. Otherwise, in some of them, you have a cell wall as well as a membrane. I'm not going to discuss more about the bacteria or bacterial wall, etc. But whenever you have an animal cell membrane, you will not have a cell wall. Cell wall is specifically typical to bacterial cell as well as a plant cell. For animal cell, you will have just membrane-like structure. So let us look at the membrane type of structure. This is a model which is based on fluid mosaic model, and it explains very nicely about the function of the membrane. We will talk more about this in the next lecture. But at this point of time, we would like to understand what this model refers to. You can see different colors, and these colors can be explained based on the structure of lipid. And this lipid structure, let me just hang on to the, okay. So now what you have is that this portion, I'm just trying to draw this portion. So this portion, so this is the three-dimensional structure. This is the membrane, one 2D membrane. So what you have is that you have lipids. So these are lipids. These are, lipid structure, and it is present as a bilayer, not single layer. So if I take this membrane, and in upper half you will have a different set of lipids, and lower half you have a different set of lipids. Same way here, you can dissect it into two forms, upper half and lower half. So I'll explain what do you mean by upper half and lower half. So this is a one half of the upper half. You will have a set of lipids which are present, and what you have is that these lipids, they are making bilayer. There is one layer which is the upper layer, and there is another layer which is a lower layer, and this is outside of the membrane, outer side, and this is the inner side. This we can say that this is the inner side of the membrane. So if I take this membrane, so how does this will look like? So if I have a cell, it should look like this, and this will be inner part of the membrane. So this is the way. Now you can also see that there are some of the thing which is inserted in this. So what are these? And in addition to lipids, there are certain things which are embedded in this. Sometimes they are present outside, sometimes it is dissecting the membrane and coming from inside to outside. So there are different forms of this blue color present. Colors are nothing but proteins. And the pink one shows you the hydrophilic part, so this head part, and the green color shows you the hydrophobic part of the lipid. So the lipid is the pink as well as the green, whereas the blue one is the protein part. Now, addition to this is a Singer-Nicolson model, which she tried to explain about the fluid mosaic. I'm trying to extract the information from here. So what you will have in some cases, these lipids, they may also have a structure like this. So if I have this kind of a structure, or it could be present in the protein. So these are the structure which are present in both lipids and proteins. And now these structures are nothing but carbohydrates. They are covalently linked either with the lipids or they are covalently linked with the proteins. So what we call these proteins? We call these proteins as the glycoproteins. These lipids are called as the glycolipids. So in addition to proteins and lipids in the membrane, you will also have lipids. So these things we are going to discuss in detail. And before we move on, let us talk about, to give you some idea that what is the thickness of these membrane. So you look at the membrane, they are sheet-like structure. They are not, circular structure or something like that. You will find that these are sheet-like structure. So if I want to draw them, they are like sheets. So they are bilayer sheets. So these are sheets. And these sheets are enclosed. So in a cell, these sheets are going to be enclosed. I'll talk about why these are enclosed sheet, but yes, these are enclosed sheets. And the thickness of these membranes, they range from 60 angstrom to 100 angstrom. That means it could be 6 nanometer to 10 nanometer in thickness. Now, as I said, that they can be boundary not only for the cell, which is a plasma membrane, which is called typically as a plasma membrane, they can be also boundary for different parts or inside the information. You also need to know what is the composition of lipid and protein. So far we have discussed that they are made up of lipids and proteins. Now question is, how much is a lipid, how much is a protein? The answer for this, it is a variable. Their mass ratio can range from 1 is to 4 to 4 is to 1. So here you may be interested to know that which one is in such a range. Suppose I'm talking about plasma membrane. What should be the composition of lipid and protein? Typically, in a plasma membrane, which has both function as a barrier as well as certain information transfer from outside to inside, or the one which can act as a transfer of a specific molecule, they have a molar ratio of 1 is to 1. Whereas some of the membranes, which has been, you know, talked about here, they are exceptional membrane. Some of them, they may have one lipid, that is lipid is very less and protein is a very high. So 1 is to 4 ratio. So which type of membrane you expect where this ratio should be present? Where lipid is less and protein is large. That means it does not require that much of a barrier function, but it has a lot of other function where the membrane proteins are involved. Very high concentration for membrane protein.
So mitochondria.
Yes, so it will be mitochondrial membrane. It could be chloroplast membrane. So these membranes, they will have a high concentration of a protein and very low lipid. On the other hand, there is another set of these membranes which has a 4 is to 1 ratio. So where do you find this 4 is to 1 ratio? Where the barrier function is extremely large, where you require nothing should be transported across the membrane.
Nuclear membrane.
No, in nuclear there is a transport which does occur. See, what is the most protected part? Our nerve tissue. So the membrane, the sheath, which is present as a myelin sheath across the nerve tissue, they are the one which will have very high content of a lipid and very low content of a protein. So this is the ratio which is present across the membrane. They may be variable. Their lipid and protein ratio may be variable. And as I said, the membrane also contains carbohydrate. And as I said, that whenever a carbohydrate is present, they can be linked to the lipids or they can be linked to the proteins. So when they are linked to the lipids, they are called as a glycolipids because carbohydrate forms glycoprotein, lipid is lipid. Same way you can call them as a glycoprotein when they are present along with the protein. And this linkage between carbohydrate and lipid or protein are covalent linkage. They are not non-covalent linkage. So that's a very important thing that they remain intact whenever they are part of the membrane. Now question comes that can all the lipid form the portion of the membrane? Or a specific lipid forms the membrane becomes a membrane component. Answer is that the membrane lipids which are they are the one which are small molecule, they are not very big molecule. They have both hydrophilic and hydrophobic portion, and these lipids spontaneously form closed bimolecular sheet in aqueous media. And that's the way the lipid bilayers are formed, and they act as a barrier for flow of polar molecule. As far as a non-polar molecule is concerned, in a small concentration, they can move across the membrane. If they are in a high concentration, if non-polar molecules are in a high concentration, what do you expect? Suppose I take membrane and put them in say chloroform or hexane. What is expected? They will dissolve the lipid portion. And if they dissolve the lipid portion, membrane will disintegrate. So at high concentration, nonpolar molecule disintegrates membrane, whereas a low concentration, they can be transported. So this is about the lipids. How about the proteins? The proteins, they are specific in nature. They can perform different functions. They can act as a pump. Pump means they are transporting molecule across the membrane. They can act as a channel. See, pump and channel, they are more or less same. I will talk about what is the difference between pump and a channel when we talk about membrane transport in next lectures. They act as a receptor. You have learned about the receptor. So the receptor molecules, they are present on the outer surface of the membrane. The ligand comes, binds with the receptor, and transfers the information. They can act as an energy transducer. They can be enzymes which are performing a specific catalytic function. So membrane proteins, if you look at the previous slide where the membrane proteins are there, these are the membrane proteins which I have shown the blue color. So where they are present, they are embedded in the lipid layer. So membrane proteins, they are embedded in the lipid bilayer, which creates suitable environment for their action. I'll explain this. Then membranes are non-covalently linked, and the lipid molecules, they are held together by many non-covalent interactions which are cooperative. Just to explain this, let us go back to the previous slide. So if I look at this lipid and this lipid, so this is the one which I have drawn earlier. So if I take these two lipids, suppose there is one lipid, I will explain what are these lipids. But if I talk about this lipid and this lipid, how they can interact with each other. These are hydrophilic part, the head group. And these are the tail-like structure. These are slightly wavy structure. So these tail structures, they are hydrophobic in nature. So what kind of a major interaction you have here? You have a hydrophobic interaction. So here in this portion, you have a hydrophobic interaction. That is a van der Waal interaction. They will keep and hold the molecule together. So the lipids, they are non-covalent assembly over here. So whenever the lipid molecule they are present, they can be disintegrated, they can again reform. So this is a very dynamic structure which is present in the membrane. Now coming to the protein part, protein also interacts with the lipid with what kind of interaction they should have? Protein? They should also have, because these are hydrophobic, so you will have only hydrophobic amino acid in this portion. So you will see when we talked about the protein structure, what we said that protein folds in such a way that the hydrophilic structure is on the outer surface, whereas the hydrophobic portion goes in the inner core of the membrane. But here, slightly different scenario will occur. The hydrophilic portion will be protruded outside, whereas a hydrophobic portion, they will be embedded inside the membrane. And that is why they can have hydrophobic interaction with the lipid. Is it clear? Have you understood the concept? The concept is very simple, that the interaction with the lipid is because of the hydrophobic interaction, that is a van der Waal interaction, and they will be cooperative because there are so many interaction which is occurring together. The lipid can also interact with the protein, and that is going to be hydrophobic interaction. Now, this lipid which I have talked about has to be both hydrophilic as well as hydrophobic. We will discuss this further. Coming to the other properties, the membranes are asymmetric in nature. What does asymmetric means? Asymmetric means that if I cut this membrane into two halves, so if I take this membrane, cut them into two halves, so here the lipid and here the lipid, and in this part the protein, and in this part the protein should be different. So they are asymmetric, they are not symmetric. That means they are different, and these lipids could be different. So these two lipids, they may differ in nature. Then okay, so what we have so far got the structure is that you have asymmetric nature and two faces of biological membrane, the one which is facing outside and another which is facing on the inner side of the cell. They are different from each other. And why they are different? Because they have a different function. So for example, anything which is moving inside should move from outside to inside, but it should not move from inside to outside. So if I have a receptor on both side, outer side as well as the inner side, then it will be like something which is a ligand is accepted outside and transported inside, and from inside again it will be pushed back to the outside. That should not happen. And that's why you have a membrane which is asymmetric. Now, can anybody tell me why membrane maintains a symmetric structure. Okay, we'll come to this point when you know how the membrane behaves. So in the next part, we have discussed about membranes are fluid structure. Lipid molecule diffuse rapidly in the plasma membrane, as do proteins. So both lipids as well as a membrane protein, both they keep on moving in the membrane, and they are unless they are anchored in the specific interaction. So there may be a possibility that some proteins they are bound to the anchor, or they are bound to the cell wall, or they are bound to the rest part of the protein in the cell. In that case, they are not movable, but otherwise they keep on diffusing rapidly in the plane of the membrane. In contrast, lipid molecules and proteins do not readily rotate across the membrane. I'll try to explain this. Membrane can be regarded as a 2D solution of oriented proteins and lipids, which you have seen in the previous slide. So in this slide, I'll try to explain. So if I have a membrane, what I said is that the lipid moves. So suppose this lipid is here right now. After a certain period of time, what you will see is that the lipid is going to, we are having pink color. Let me select gray color. So suppose lipid is here. I have chosen wrong. So suppose lipid is here, after a certain period of time, the same lipid can reach here. That means it is a movable, it keeps on moving. Now, unless there are certain, anchoring proteins which are bound to this or lipid, which on the inner side, they will always be movable. So at a given time, what you are looking at here is, it is showing a model which shows that in sea, there are certain icebergs which are floating. So if I consider these ones, they are iceberg, and this is the sea. The pink color is showing the sea. So what is happening is that these icebergs, they are floating. So this is a typical fluid mosaic model. Do you understand what is a mosaic? Mosaic means these structures, they are scattered and they are not the same structure all across. So this mosaic of protein present, so these proteins, they are embedded. It is not showing in three dimension, but they are embedded inside the protein. These are also embedded. Now, so first thing is that these proteins, as well as, so this protein, for example, this protein is present here. After a certain period of time, they may reach here, and here you will have protein. So this is the possible when you have non-covalent assembly of lipids and proteins. So they are not bound by covalent bond. Otherwise, you will require a lot of energy for their movement. You will require a lot of energy for their transfer. Second thing which we have said is that the lipids or proteins, they do not move from outer part to inner part. So suppose there is a lipid here, it will not move here, it will not come to this side, or the lipid over here will not move outside. That's one thing, and that is why what happens, the membrane is asymmetric. That means this portion lipid remains whatever it was, and this portion lipid remains whatever it was, though there is a movement of lipid in the one half of the layer. So suppose this is the half of the layer, this will keep on moving. The second half, they will keep moving, but they will not move from this half to this half. Is it clear to you?
Can you please repeat this portion?
See, what I'm saying is, suppose I have a membrane, and in this membrane, there are two halves. Suppose this is one half, and there is another half. So these two are two layers. So we call it a bilayer. So this is two layers. Though in the membrane, I'm not going to say, this is the distinction line which shows this is the outer layer, this one is the outer layer, this is the inner layer. So what I'm saying is that this lipid and protein, they will move in a particular layer. So if it is in an outer layer, they will move in outer layer. So this can move to here, right? Whereas this lipid can move from here to here. Same way, a protein which is present here can move from this portion to this portion. But what I'm saying is that if suppose there is a protein present here, outer membrane, this will not move to inner membrane. So movement within the layer is a possible, but they don't move from one layer to another layer. That means it is not moving from outside to inside or from inside to outside. Why it is possible? Is it possible? Why it should happen? And how it is being maintained? That means how asymmetry is maintained in a membrane. It is maintained because what happens within the layer, when it is moving from this layer, this part to this part, they do not have lot of energy restriction. But suppose they are moving from this part to inner side, what you have? What is the barrier? Barrier is again lipid molecule. And if I look at the outer surface, this is the hydrophilic one. So this hydrophilic has to cross the barrier. And if hydrophilic molecule has to cross the barrier, which is hydrophobic, this will cost energy. And that is why most of the time, or 99% time, these are not moving. And if any such movement occurs, it is known as a flip-flop movement. But this does not happen except in exceptions, like in virus membranes, some of the enzymes present, they help, or they help in the reducing the energy barrier, that is possible. But in general, the movement across the membrane, either for lipid or for proteins, does not happen. Okay, let's move on to
So,
the next concept. The most of the cells, they are electrically polarized. So suppose I have a membrane, on inner side you will have a negative charge, and that is having a membrane potential which is a typically minus 60 millivolt. This kind of charge distribution is happening because of certain transport. We'll talk about that. But yes, this is what is the characteristic of the membrane. Now, looking at the membrane, we have talked about the lipids. The major component of the lipid, which we have talked about. So suppose I have talked about a structure. Let me again draw the structure. This is always you have to understand this is a very short form of lipid. So in this structure, this chain, whatever I'm referring to, this chain, they are nothing but fatty acid. And the upper part is the hydrophilic part, but these are fatty acids. So fatty acids are constituent of lipids. They play an important role in signal transduction pathway. This part we will talk about when about the signal transduction, almost at the end of the course. But yes, the lipids, they are not esoteric molecules. They are not the one which are just acting as a hydrophobic barrier. They are also involved in very important function like signal transduction. We will talk about that. Then hydrocarbon chains of various length and degree of unsaturation with carboxylic group terminating, they are the one which are making the fatty acid. So if I want to define fatty acid, what are the fatty acids? They are hydrocarbon chain of various length and degree of unsaturation. They can be unsaturated, they can be saturated, as well as at the terminal point, they will have a carboxylic group. This is what you have already learned in 11th class and 12th class. And if I want to name them, again, their systemic name is derived from the parent hydrocarbon chain. So suppose I have a 18 carbon, what we call them, any alkane with 18 carbon is known as octadecane. And if I have a fatty acid having 18 carbon and the terminal OH group, they will be called as octadecanoic acid. That means from decane, we remove E and put oic acid. So from octadecane, I have removed E and put oic acid. So it's a very simple nomenclature, but you will find that there are some trivial nomenclature which is also being used. So that is what is being given. So the position at which there is double bond, that is also to be named. Then coming to the fatty acids, which are, they are the one which are very important for the physical property of the membrane. So we will talk about them. So the property of the fatty acid and lipids derived from them are dependent on their chain length and degree of unsaturation. So these physical properties dependent on how small or how big chain you have. And second thing is that how many double bonds and where these double bonds are present. And not only that, you also need to know whether it is a cis or trans double bond. So mostly in most of the fatty acid, you will have a cis double bond, and that is what we will talk about. Now, there is a very typical thing which you will learn or you may have already come across in organic chemistry, where unsaturated fatty acid have a lower melting point than saturating fatty acid of the same chain length. So suppose I have a two fatty acid, both having 18 carbon. The one which is having the double bond, they will have a lower melting point than the one which is having which does not have double bond. Can you tell me any example of this? Take this example from your kitchen. If you have a saturated fat, what happens? During this winter, they will form a solid structure. You call them ghee. So they are transformed into the fat. Whereas in winter, the unsaturated one, they will not, make fat. So what is happening? It is dependent on their melting point. So if we have a saturated fat, that means they will require more temperature to melt. Whereas if I have unsaturated fat, they will require less temperature to melt. So that is what defines what is their status, whether they're a solid or liquid at room temperature. So most of the nowadays what you eat, you take mostly unsaturated fatty acids or PUFA. Have you heard of PUFA in your oil? Any one of you? So many times when you get oil, some of them they say contains PUFA, PUFA, which means polyunsaturated fatty acid. So these polyunsaturated fatty acid, if it is present in the oil, they will, they will allow them to, solidify at quite high temperature, and that's why, sorry, very very low temperature. I'm sorry. The solidification occurs at very very low temperature, and that's why most of the time at room temperature they will be in the liquid or oil form. Now, coming to the physical chemistry of this, why should a small chain or the presence of double bond affect the physical property of fatty acid or lipid? Anybody? If I have a small chain, they will
So
have
it
yeah,
affects the van der Waals interactions between the hydrophobic parts
yeah.
of the.
So if there are more van der Waal bonds, you require more energy for them to melt. So a smaller chain will be more in liquid, whereas a longer chain will be more in the solid form because of the high interaction. How about the presence of unsaturation? Whenever there is an unsaturation present, you can see there is one example given of oleic acid. Whenever one unsaturation is present, I will show you some more, structure of this oil or fatty acid. You will see there is a kink which is present in the lipid, and because of the kink, packing becomes loose, and that is why whenever there is an unsaturation, the membrane remains in the fluid state. So short chain and unsaturation enhances the fluidity of fatty acid and their derivatives. So when you talk about the membrane, a membrane having more unsaturated fatty acid will be more fluid. The one which is having more saturated fatty acid will be more solid. Is it clear? Now, the types of membrane lipids, one of them, very common one, is that is phospholipids. I have given you a definition. The lipids are water insoluble biomolecules and are highly soluble in organic solvents such as a chloroform. These lipids, they are fuel molecule, concentrated energy storage, signal molecule, component of a membrane. Then we have also talked about that there are three major kind of membrane lipids. One of them is a phospholipid, then glycolipid, and the cholesterol. Now, at this point, I have a question. Suppose I am giving you a structure. This is the structure. I am trying this as a glycerol chain. So you have CH2, OH, CHOH, CH2OH. And here I am having R group. So this is what I am trying to have. So here RCOH, RCOH, RCOH. So there are three fatty acid which is attached. Say we number it 1, number it 2, number it 3. So there are three fatty acids linked to a glycerol chain. And to name it, I will name it as a, how should we name it? We should call it as a triacyl glycerol, because three position you have acyl groups and which is linked to glycerol, so it is called as a triacyl glycerol. Tell me whether it will form a membrane structure or not. So for forming a membrane structure, the thing you require is that they should be both hydrophilic as well as a hydrophobic. Now in this structure, what you have, your exposure is only for hydrophobic. So these are fatty acid R1, R2, and R3. These are the fatty acid. So this is exposing to hydrophobic. So if I take such a structure, put them in a beaker of water, what will do? They will form a bigger droplet. They will form a bigger, structure, and they will make a kind of storage molecule. So these are tricyl glycerol. They are mostly for a storage molecule. They are not part of the membrane. But now, out of this structure, two of them, we still have RR. So here I have R1, here I have R2, and here I have put a structure, say phosphorus, where I have a negative charge. So you have O, then you have OH, here you have double bond, here you have OH, here I have O negative. Now, how about this structure? This structure will have a hydrophilic structure as well as a hydrophobic structure. So this part becomes hydrophobic, this part becomes hydrophilic. Now, I'll show you further these structures. So these hydrophobic structure will help them to go away from the water, whereas this part will make them to act or interact with the water. So these structures are shown as typically a head group and tails. So these tails are nothing but these two chains of fatty acids, and this head group is nothing but this phosphate group. Okay, so if I have this kind of a structure, they can become part of the membrane, whereas if I have a triacyl glycerol, which is just hydrophobic, will not form the part of the membrane. Suppose I just have fatty acid, they will not become the part of the membrane. But if I have phosphoglyceride, they become part of the membrane. So phospholipids are the major class of the membrane lipid. A phospholipid molecule is mainly consists of fatty acids, a platform to which these fatty acids are attached. So I have shown you which platform, a phosphate residue, and alcohol attached to the phosphate. So these are the components. I'll just show you in the next slide how this works. The platform on which phospholipids are built are glycerol. So I have shown you the glycerol was there on which two fatty acids are attached, and at one position you will have phosphate. It can also be linked with the sphingosine. That's a complex alcohol. Now phospholipid derived glycerols are called phosphoglycerides. Glycerides is coming from glycerol, phospho is a phospholipid. Then sphingosine, if it is derived from sphingosine, which is another alcohol, it is called as sphingomyelin. Now, look at this structure. This is the one which I was trying to draw and give you a complete structure. So you have a glycerol. Can you see glycerol here? In this structure, what we have is we have glycerol, and so this is the one which is a glycerol. It is the backbone. Now, there are two fatty acids. These fatty acids are hydrophobic in nature, and this part becomes hydrophilic. So this molecule has both hydrophobic as well as hydrophilic, and when we draw it in a symbolic structure, this is the way they are given. So these are the head. This head group represents this part, and these tails represents these fatty acids, two fatty acids chain. Now coming to the typical structure here, how the glycerol has been shown, you have this carbon having R1, first fatty acid. So you have, this is the glycerol chain, CH2OH, CHOH, and CH2OH. So this first two, the fatty acids are linked. You can see R1 and R2. And the third one, this the third one, this carbon, you have a phosphate group attached. Now this phosphate can also attach with the alcohol. And these alcoholic groups, which were trying, are different. This alcoholic group could be serine. Have you heard of serine somewhere? Do you
It's
remember
a
serine?
it's
This is
a
one of
Yes,
the
amino
sir.
amino
acid.
acids.
Amino acids.
Yeah, this is one of the amino acid. But what you have is that it has also OH group. So what will happen? This OH group will interact with the OH group of phosphate, and that is what will form a serine bound to phospholipid, and that is what is called as a phosphatidyl I will show you this structure. Same way ethanolamine. You look at this structure. What you have is that you have, instead of CH3 CH2OH, one of the H has got amino group. So it becomes ethanol amine. So ethanol is C2H5OH. You have attached one amine. In choline, what has happened? That the amine group of ethanolamine has further got three CH3 groups. So all the H has been replaced by CH3. Of course, this is a typically a glycerol. Then this is inositol. This is like a glucose, but all of the is modified into OH. So it's some having polyhydroxy alcoholic structure. So all of them, they can become part of the phospholipid, where I said alcohol. So this alcohol can be so this alcohol which I was talking about could be any one of them. So this alcohol could be choline. This could be ethanolamine. This could be serine. So what we'll call them? If there is a choline, we'll call this a phosphatidylcholine. If it is ethanolamine, we call it as a phosphatidylethanolamine. If it is a serine, we will call it as a phosphatidylserine. If it is inositol, we call them as a phosphatidylinositol. So that way your phospholipids are named. And so typically the same structure is there, but the modification in alcohol occurs. In the next slide, you will see all the structures. So all these structures I'm showing you because you will be doing your experiment when you come to the lab, and you will be isolating all these structures. So you look at the first structure. Here, what you have is that this red color, whatever is written, is nothing but serine. You have amino group, CH, on one hand COH group, other side you have CH2OH. So this is a serine. This is a choline. Now choline, what you have is that similar structure like ethanolamine, and nitrogen has got three CH3 group, which are replaced by, instead of H3, it has got three CH3. Phosphatidylethylamine, same. Instead of having CH3 here, one of H has been replaced by amine, so we call it as a ethanolamine. Phosphatidylinositol, the same group which I have shown you in the previous slide, has been attached here. So all of them, they are present as a part of the membrane structure and membrane lipid structure. And why it is feasible for them to form a membrane? Because the structure which is in the red color, they should act as a head part of it. And these fatty acids, these ones, they are the one which are depicted as the tails. So you have a head which is formed of the phospho part, which is a component of one of the alcoholic group, which is a hydrophilic in nature, and these are the hydrophobic. So the interaction of these chains are because of the hydrophobic interaction, and the interaction of the head group is because of the your interaction with each other, as well as this will also be stabilized because of the water which is present on the outer surface. So at this point, I will stop because time is over. We'll continue with this in the next class. Any question if you have?
Sir, I have a question.
Yeah.
So the proteins in the bilayer,
Hmm.
actually, sir, since the movement of the proteins is also allowed in the layer, they could
Right.
aggregate, right, due to hydrophobic interactions between them.
See, that is what happens. There is a dynamic they each other. Will not say that they collapse, but they can meet with each other. So suppose, and that is the beauty of the control. Suppose I say that a protein can become functional when two of them interact with each other. So what will happen? There may be some trigger, and that trigger comes from the receptor. So suppose a protein has been receptor. Their ligand comes, binds with them. Then this protein moves within this fluid structure. They interact with another protein, and then the second protein becomes a functional protein. So that complexity I will tell you when we talk about the signal transduction. And in fact, some of the function of the membrane has been given based on the fluidity of the membrane. If membrane is made rigid, membrane will not work. And that is what has been found. So what happens, suppose I take a membrane, and then if I want to make them rigid, can we make it? Can you tell me some strategy where I can make the membrane not being fluid?
So we could increase the like more amount of saturation would make it like rigid.
Okay. But that is not enough. Very simple thing could be that I take any membrane and put them in a fridge. So at 0 degree, the movement will be lost because all the lipid will become solid, isn't it? So lowering the temperature. So here we are following another physical property. That is, lowering the temperature will solidify them. And once they become solid, they will not work. On the other hand, if I heat them above 40 degree, most of the membrane, they will become totally loose. They will disintegrate. So, so whatever you have asked as a question, it is very valid in the biological system. And instead of increasing and decreasing the temperature, what you see is that it is the interaction within the protein that makes them active protein. So some of the protein they work, and in most of the hormone interaction with the membrane has been explained earlier based on their interaction and movement of these proteins within the bilayer. Any other question? So you keep on having these questions because these are very important, and that's what is the logic of understanding the biology. Okay, if there are no more questions, I will sign off. Thank you very much.
Thank you, sir.